Hello students, today now we are going to discuss about genome organization in plants and also gene expressions. So before going that one, sir, where we are looking the genes, what is the meaning of genes? So generally now you see, so when you are looking in the cell, center part of the cell we call that is a nucleus. So this nucleus is contain the DNA. So we call that is genetic deoxyribonucleic acid. Similarly, one more is there, that is in RNA, ribonucleic acids. So these DNAs contain some type of the codes. So we call that is in genes. So actually what is the meaning of genes? Gene means it is a one type of the liquid materials. So these liquid materials gives to informations is goes to one, so life to another life. So you see sometimes the genes you are looking, so when it is so different type of the structures you are looking in the genes, the gene has expressions in only at the time of, so when we are looking in the so future or any times, so that is often gene expressions. So now, so before going that one, so our senior professors, so Dr. Associate Professor Mr. PADALA Thirupathi sir is clear explain so about the what is the gene and what is the gene expressions. Sir, please, so what is the meaning of genes? So where we are looking in the genes, how the genes are expressions. So please give me the details about on that one, sir.
Thank you, Ramakrishna sir. Actually, gene is the structural and functional unit of heredity. We know that. So today we are going to discuss about genome organization in plants at the same along with gene expression. So gene, it is the molecular language of genes. Genome organization refers the structural functional arrangements of genetic material within a cell. In plants, the genome composed of nuclear DNA, it is a linear DNA, it is acted as a actual genetic material in plants, including all living organisms also. Along with this nuclear DNA, some of the other DNAs like plast DNA, that is we can also call as chloroplast genome or CP genome or CT genome, and mitochondrial DNA, it is very important in maternal inheritance that we can also call as mtDNA. These are all along including genome, nuclear genome, these extranuclear genome is also present in living organisms. The nuclear genome is the most complex housing genes responsible for plant growth, development, metabolism and adaptation. The genome organization in plants varies widely among species due to different genomes, size, ploidy level and repetitive DNA content.
Actually, the genetic material we are looking in only center part of the cell.
Yes.
Yes. So just center part of the cell means where the chromosomes is there. In that chromosomes we are looking the genes or genomes. But here what happens are sometimes the genes mean this genome we are looking outside the center part of the cell.
Yes.
Mean the gene we are looking in chloroplast, the genes we are also looking to the mitochondria.
Yes.
That's why when we are looking in genes in mitochondria, we call that is in mitochondrial DNA.
Yes.
Similarly, second that is of in chloroplast DNA.
Yes.
So what type of the functions we are looking in, the same type of the functions we are looking in mitochondrial DNA or CPD central DNA, or any similar or structures? What is the differences between central genome and so both mtDNA and ctDNA, sir?
Yes, actually DNA's main function of the DNA is acted as a genetic material.
Okay.
But along with this nuclear DNA, the chloroplast DNA and mitochondrial DNA, it is quite different from nuclear DNA. Why? Because nuclear DNA is a linear DNA, but in chloroplast or mitochondrial DNA, it is a circular DNA. These two DNAs are functions are quite different from nuclear DNA. Chloroplast DNA, it is responsible for the production of the enzymes which are responsible for the photosynthesis, and also it involves the maternal inheritance mitochondria in aerobic respiration also. But the nuclear DNA is actual genome, actual DNA for hereditary process in all living organisms.
Yes, correct, sir. Means where the center part of genome is that is in only most important.
Yes.
Yes. Continue,
Then the components of the plant genome, nuclear genome contains, in the center part of the, what Ramakrishna has told, sir already told, the center part of the nucleus, it contains the chromosomes, the gene coding sequences, regulatory elements and repetitive DNA and non-coding RNAs. These are all involved in the nuclear genome. Chromosomes, we know that chromosomes are the most essential, most important process for the all type of the cell divisions, mitotic cell division or meiotic cell division, whatever it may be, whether they are the autosomes or whether they are allosomes, whatever it may be, but the chromosomes are responsible for the exchange of the genetic information from the parents to their offsprings in meiotic cell division. But the best examples of the plants, that is chromosomal plants exhibits varying chromosome numbers in Arabidopsis thaliana. This is the best suitable plant for the all type of the advanced research methodology. The Arabidopsis thaliana is the best suitable plant. This plant has consist 5 chromosome pairs, while wheat has 21 pairs. This is the varies of the chromosomes in different plants. The chromosomes consist of the chromatin. It is a dark red dark thread. It is a gene rich actively transcribed regions, and heterochromatin region, that is very gene poor transcribed region we are observed. Gene and coding sequences, protein coding genes, 20,000 to 40,000 in most plants we are observed, are organized into exons and introns. We are already discussed in that exons and introns in the process splicing process, introns are removed and exons are only joined in the process of the post translation methods. Here the regulatory elements, promoters, enhancers and transcriptions factors which are involved of the regulation of the gene. The repetitive DNA, it contains tandem repeats and transposable elements. These transposable elements also we can call as the jumping genes, which are discovered by Barbara McClintock. The non-coding RNAs here, the microRNAs or the miRNAs, long non-coding RNAs, that is the INC RNAs or long chain RNAs, that is which regulate gene expression post transcriptionally. Here I am showing here gene organization in prokaryotes, whereas in prokaryotes only the that becomes, there is no definite nuclear membrane. Without nucleus, only the nucleoid region which we are observed, that is the genome which we are called as nucleoid. So it is only present in prokaryotic organism like the bacteria or cyanobacteria. This is the acted as a genetic material. But whereas in here, the right side image, what we are observed in the nuclear genome, chloroplast genome and mitochondrial genome. Chloroplast genome and mitochondrial genome, which we can call as extranuclear genome. But the nucleus genome is actual genome which is acted as a genetic material all eukaryotic organisms. The plastidial genome or plastidial DNA, it is a circular DNA which present in chloroplasts, especially the circular DNA which is involved in the photosynthesis, synthesis of the different type of enzymes which are involved in the photosynthesis of the stroma of the chloroplast, that is circular DNA. But the mitochondrial DNA which is present in the matrix of the mitochondria, it is also a circular DNA which is involved in the maternal inheritance. It is very, very important, my dear students, remember maternal inheritance, the best example involved, the best example cell organelle, that is mitochondria. Mitochondrial genome which is also involved in the respiration, especially aerobic respiration. The most suitable enzymes are synthesized by this mitochondrial genome. Next, then what are the organelles genome? We'll discuss in the previous slide. Here I am showing about the chloroplast genome. Typically it is less genes are present compared to the nuclear genome. It is a double stranded circular DNA and it contains 120 to 160 kb nucleotides are present, and it encodes proteins involved photosynthesis, that is especially ribulose 1,5-bisphosphate, carboxylase and oxygenase enzyme. It is abundant enzyme. It is a dimorphic enzyme which is only present in the chloroplast DNA, and has conserved regions including the large single copy, that is LSC region, and it contains small single copy, that is SSC region, and inverted repeats. These three parts which we are observed in the chloroplast DNA. Along with this mitochondrial DNA, large and more variable size 200 to 2400 kb nucleotides which we are present in the mitochondrial genome compared to the chloroplast DNA, and this encodes components for oxidative phosphorylation and production of the adenosine triphosphate, that is ATP. It is also we can call as the assimilatory power in the photosynthesis, even though the respiration, but undergoes frequent recombination and rearrangements by the involvement of this mitochondrial DNA. And the genome complexity is also most important in different plants. Arabidopsis thaliana, the best example of the plant we are discussing here, more than it is 135 MB to extremely large, that is Paris japonica. It contains 150 GB. 135 MB is quite differ from 150 GB. It is very larger compared to these two different plants, the plant genome size range. And genome size does not directly correlate with organism complexity, that is C-value paradox. Here polyploidy whole genome duplications is common in plants and plays a key role in evolution of the species, evolutionary process. And along with this extranuclear genome, some of the chromatin organization and epigenetics we will going to discuss here. In this, histone proteins, these are the most stable proteins, very important proteins which are involved in hereditary process, where all the chromosomes DNA contains these histone proteins. These are the basal proteins which involve the transform the genetic material or genetic information for genetic makeup by the involvement of these histone proteins.
Above the gene, we are looking the proteins.
Yes.
Yes, sir.
Yes.
So means here when you are looking the histoproteins.
Yes.
So we call that is of nucleosomes. So what is the histoprotein, sir?
Here we are discussed in a nucleus. Eukaryotic organisms contain two types of the proteins in the nucleus. One is basal proteins, one is acidic proteins.
Okay.
So that is, we can also call as basal proteins, we can call as histone proteins. Non-histone proteins are acidic nature, that is not showing the basic nature, alkaline nature. So this basic nature, alkaline nature, is suitable for the transform of the genetic information from parents to their offsprings. Here the histone proteins are involved. DNA is wrapped around the histones to form nucleosome. Nucleosomes are nothing but a beaded like structures which are arranged on the thread like structure of the chromosome. The compacting into chromatin. So that is nucleosome. It nucleosomes which shows nucleosome actomer. Acto means eight histone proteins which are involved in the inside the nucleosome, and the another one protein, that is H1 protein, histone 1 protein, is present on the surface of the two nucleosomes that we can call as linker DNA. So H1 protein is present on the surface of the, or it is involved in the linker DNA. Two nucleosomes are connected with this chromatin thread. In that place H1 protein is present. Remaining histone octamers or 8 proteins are present inside the nucleus. That nucleosome that we can call as nucleosome octamer. Histone octamer. This nucleosome is discovered by Audet. Audet was the scientist who discovered and clearly explained about the nucleosome. Here the second point we are discussing here, DNA methylation. Without DNA methylation, there is no gene expression. Remember here, regulator of the gene expression and silences transposons. This is just because of the DNA methylation. Along with this histone modifications, by the influence of the chromatin accessibility and transcription, histone modifications which we are observed inside the nucleus. And then what are the functional aspects of the genome organization? Before that one, structural aspects. Now we are going to discuss about the functional aspect of the genome organization. In this genome clustering process, these genes with related functions are often clustered together. The best example, the secondary metabolites biosynthesis genes. Genes are involved synthesis of the secondary metabolites like alkaloids, tannins, resins. These are the different types of the secondary metabolites which are synthesized by involvement of the gene clustering process. Operon and co-expression. Some plant genes exhibit operon-like structures. Most of the prokaryotic organisms like bacteria or Escherichia coli, this bacteria shows different operons, lactose operons, lac-operon and tryptophan operons. We'll going to discuss next slide. Here genome rearrangements, duplications, inversions, recombinations and drive genetic diversity. Then the conclusion of the genome organization here, the plants is highly dynamic, shaped by evolution, environmental adaptation, genetic diversity. Advances in sequencing technologies continue to unravel genome complexity, providing insights to plants biology, crop improvement and conservation genetics. Now we are going to discuss about the gene expression. Gene expression is the fundamental process by which genetic information encoded in DNA and transcribed into RNA then translated into proteins. This process is essential for plant growth, not only plant growth, any type of organism. The development of the organism or the growth of the organism, it is controlled by this gene expression. The developmental, even though metabolism, response to environmental stimuli, gene expression is highly regulated at multiple levels, ensure the proper functioning and adaptations. Then here we are going to discuss about the different types of the gene expressions. Number one, that is constitutive gene expression. Next, regulated gene expression. This regulated gene expression, it is more complexity compared to the previous one. Here the categories of the regulated gene expression here, inducible gene expression, that is the best examples of the lac-operon. It is we'll going to discuss here. Then tissue specific gene expression, repressible gene expression. Repressible gene expression is the best example, that is tryptophan operon, lac-operon. It is we'll going to discuss next slide. Then developmental stage specific expression. So developmental stage expression, the best suitable plant, that is Arabidopsis thaliana, the leaf and even the flower pods, that is apetala one, leafy cotyledons. These are different types of the segments which we are discussed in that developmental stages of the specific expression. And stress responsive gene expression. Here stress is quite differ, that is if the stress is caused by organism, that we can call as biological stress. If without involvement of the living organism, the physical process like temperature, salt, water, drought, these type of stresses we can call as the abiological stresses. In this type of stresses, how to control, how to avoid, how to sustain, how to cope the stress, it is controlled by this gene expression. Due to this cause, if the number of days plants facing the floods, even though the plants can sustain just because of this gene expression or regulation of the gene expression, even though large number of days plants are sustained during hot summer in the desert, even though that is sustained just because of this gene expression. Then the molecular mechanism of the gene expression. So here, then after structural and functional aspects, now we are going to discuss about the molecular level of the mechanism of the gene expression. Here the molecular level of the gene expression will discuss here transcriptional regulation. In this transcriptional regulation, the binding of the DNA sequences promoter regions, even though the cis regulatory elements that is abscisic acid responsible events that we can call as ABRE, or after transcription, then post transcription regulations, how microRNAs, how small interfering RNAs, siRNAs, how involved in this mechanism, then translation regulation and post translational modifications. These are all involved in the molecular mechanism of gene expression. Next, even afterwards, then structure of gene. Then gene contain different components, that is regulatory region and the coding region and terminator region. Any gene contains these three regions. The regulatory gene, that non-coding region, contains promoter, enhancer, silencer along with operator. Then coding region, that we can call as exons. The exons is only coding regions which encode amino acids, whether suitable amino acid, even though essential amino acid, whatever it may be, but they can code amino acids forming the functional protein, due to involvement of the coding region. Then introns, these are all called non-coding sequences which is removed by RNA splicing. We are already discussed in previously. Then RNA splicing is most important for the removal of the non-coding sequences, that is introns. Then last, terminator region of the gene. The terminator region is most important. Whenever the coding is the transcription is going to end part, or the signaling to end the transcription, that is terminator region. Then what are the properties of gene? Which type of properties show all genes? Any gene can show a specific properties that we can discuss here. The specificity, collinearity, mutability, regularity, regulability, heritability. These are the specific properties of the gene. The first one, specificity. Each gene, any type of gene, each gene codes for a specific protein or even though RNA molecule. That is, we can call as specificity of the gene.
Here the gene means it is one of the liquid materials. Every gene is contains separate particular code.
Yes.
One gene is not to the same other genes. One gene to other gene, lot of variation is there.
Yes.
Yes, sir.
Yes, definitely.
Yes, sir.
So that's why each gene is their own specificity, shown their own specificity. The best example, AUG codes for methionine only. That is the specificity. And at the same time, UAG codes for not code any type of amino acids. So that's why they can call as termination codons. So that is also a specificity of the gene.
Okay,
and collinearity, the second properties of the gene, collinearity. The sequence of nucleotides in DNA corresponds to sequence of amino acids in a protein. Then mutability, genes can undergo mutations. Mutation is nothing but a sudden changes occurred in the gene sequence or genetic sequence that leads to variations or different genetic disorders. And regulability. So gene expression is tightly regulated by internal and external signals. Heritability. This is most important to transform from the genetic material from the parents to their children or their offsprings. Now we are going to discuss most important criteria, the hypothesis, that is one gene, one enzyme. What Ramakrishna sir has already asked me previously, one gene code for one enzyme. It is given by Beadle and Totem in 1941 in Neurospora crassa. It is a fungal member, and this gene codes for only one enzyme.
Yes, sir, this one is the most important here. Means one gene is contained particularly special type of the code.
Yes.
So these type of the code we are not looking to any other type of the genes.
Yes.
Yeah, that's why one gene is contained particularly special type of the code.
Yes,
sir?
that we can call as specificity.
Specificity, yes,
Yeah.
sir, yes, sir.
Yes, sir.
Please.
Then in this hypothesis, some of the limitation is there. Why? Because not all enzymes code, not all genes code for enzymes.
Okay.
Some codes only proteins, some are regulatory RNAs. So that's why this is not acceptable. Afterwards, after this hypothesis, The another hypothesis has given, that is one gene, one polypeptide hypothesis. This is also we can call as modified hypothesis. It has given by Ingram in 1958. So in this research, polypeptide. So one gene codes for polypeptide. The best examples of the human hemoglobin consist of 2 alpha globin and 2 beta globin polypeptides, each coded by separate genes. Then this gene is also not a single part, it is a divisible. This phenomena is practically evidentially proved by Seymour Benzer in 1955 in bacteriophage T4 type of bacteriophage. This according to Seymour Benzer, gene is divisible. It divides into 3 types, that is recon, mutant, cistron. Recon is nothing but recombination unit. Mutant is nothing but mutation unit. Cistron, remember my dear students, cistron, this is actual gene. Cistron nothing but actual gene. It is a functional unit. Okay. So what are the significance of this process is research realized that divisible and not the smallest unit of heredity. Next, we are going to discuss about the gene expression in the control, how to control mechanism of these metabolic processes. Number 1, lac operon model, that is inducible operon. It is given by Jacob and Monod 1961 in the Escherichia coli bacteria. This lac operon structure contains regulatory gene, structural gene,
Yes.
promoter, operator gene. Structural genes contain LAC-Z, LAC-Y, LAC-A. The mechanism of this lac operon, in the absence of lactose, the operator binds the operator, repressor binds to the operator and blocking the RNA polymerase, automatically transcription is stopped. If in the presence of the tryptophan, the repressor binds to the repressor, activating it, the repressor binds to the operator, then transcription is blocked. This is the operon model in lac operon, and this is the tryptophan operon model. Then the conclusion, gene expression is highly dynamic. It is not a static. It is tremendously changes. It shows tremendous changes and tightly controlled process in plants and essential for growth and development and environmental adaptations. Why? Because so many changes has occurred in the environmental process, in external environment. We can understand these mechanisms allows for advancements in plant biotechnology, crop improvement and stress tolerance engineering. Why? Because in present scenario, rapid development of the population, rapid development of the urbanization process, these crop improvement and stress tolerant plants is most important for the breeding process and giving the food, and it control the hunger process, hunger. So that's why in agriculture and horticulture and floriculture and different type of the technologies is involved in this gene expression level.
Okay, sir. Nice explanation and elaborate explanations. So very much thank you. Sir, here the gene expression means it is a very much helpful to the plant biotechnology and crop improvement and stress tolerance in engineering departments. Sir, Enu Tirupati sir, nice explanations. What is the genes and what is the gene expressions? Thank you very much, sir.
Thank you, sir. Ramakrishna sir.